Okay, so today's video is going to focus on those last few techniques for genetic engineering, okay, and ways that we manipulate genes. Last time we talked about transgenic organisms and the restriction enzymes, and so we're just going to continue with that with this video. So the next one we have here is reproductive cloning, okay, and so reproductive cloning is exactly what you would think it is. We're making an exact copy here of an entire organism, okay, so not really just a cell here. We're making, it can be a cell, but we're also talking large scale here, an exact copy of the organism. The example we see over here on the side is the lamb Dolly. Okay, remember when, well, I guess you guys are probably too young, but this was big news when they did this, when they cloned this sheep for Dolly. Okay, they got an egg cell, and when they cultured the egg cell, and then the egg cell developed into the embryo, which they implanted into a surrogate mother, okay, and they got a genetically identical organism as the original donor. So far, we've been able to clone things like DNA. Okay, we've been able to clone cells, tissue lines, stem cell lines, entire organisms like the sheep that you see there. Okay, we've been able to clone plants. Okay, some animals are able to clone themselves. Okay, y'all remember reading that article on parthenogenesis? When we did meiosis with the sharks, didn't fit. Okay, and remember this was a asexual form of reproduction. All right, squeezed in there. Okay, so this is an asexual form of reproduction that occurs in nature, where you have offspring that are born via sexual reproduction, and then they reproduce asexually. Okay, so those are all, like I said, just types of cloning, but we're making an exact copy of an organism. So like I mentioned in the last one, we need to be thinking about our ethics. Is this okay? Is it okay to clone cells and tissues? Is it okay to clone stem cell lines? Is it okay to clone an entire organism, or is that the line? Smaller than that's okay, but that's too far. Okay, like I said, we'll be talking about this some more in class. Okay, so the next technique is reprogramming cells. Okay, so taking the cells and reprogramming them into a different kind of cell, and this is going to involve the use of stem cells. And so the benefits to this would be things like organ and tissue regeneration. I can fight a number of diseases to be able to reprogram these cells: cardiovascular disease, Parkinson's disease, where the cells don't produce enough dopamine. So if they can reprogram those cells to now produce dopamine again. Okay, Alzheimer's disease, leukemia, sickle cell anemia. So this involves the reprogramming of these stem cells. And the stem cells that work best come from embryos. And this is a hotbed issue. This is a nationally debated, huge issue with funding. Will they even fund things for this? Because there's a lot of resistance and pushback to this. And there is a lot of misconception that goes along with this stem cell research. First and foremost being that stem cells that came from embryos came from aborted embryos, which is completely untrue. There's tons of regulations in place as to where the stem cell lines come from. There was a long period of time where they weren't allowed to create any more stem cell lines, and so they could just basically clone the ones that they had. But they do not come from aborted embryos. They come from couples that have chosen to donate extra embryos that they've done from through fertility treatments. And when you go through fertility treatments, they create and freeze a number of embryos because it may take multiple tries and you implant more than one at a time. Okay, and so all of those embryos were essentially never meant to be implanted in the first place. And so some couples would choose to donate those for research. So they do not come from abortions. Okay, so let's talk a little bit about how a stem cell would work. Okay, so we have basically two kinds of cells in the beginning. Okay, we have a totipotent cell, which we've talked about before. Okay, remember, totipotent cells have the ability to develop to within any cell in the organism. They can give rise to an entire organism. Plant cells are totipotent. You can see here in this picture with the carrots, they take little pieces of this carrot, okay, just these little pieces of the carrot here that can be cultured, okay, and they will develop into a whole new carrot because plant cells are totipotent. Stem cells are undifferentiated, okay, but most of them are pluripotent, which means they can turn into most cells. Okay, they can give rise to most types of tissues, but not an entire organism. Okay, and so in animals, the zygote here, okay, so the zygote is totipotent. The embryonic stem cells, which are these cells in the inner mass here of the embryo, they are pluripotent. And again, pluripotent can give rise to most tissues, but not necessarily the whole organism. And so the thought process behind this research would be that we could manipulate these stem cells to differentiate into whatever was needed. More nerve cells, blood cells, liver cells, whatever particular cells were needed, and then implant those into the recipients. Let's look here at what is called an iPS cell. Okay, so an iPS cell, this is an induced pluripotent stem cell. And so what this is basically these are reprogrammed genes that are spliced into a normal human skin cell. So for instance here, or a human somatic cell. And so this instance here is a human skin cell. Okay, and so it's got the DNA that's already in the skin cell, and then there are reprogrammed genes which are spliced into the virus. And so the virus, remember, viruses will incorporate their genomes into their host cell. And so the virus injects those genes into the host cell, and we end up with a recombinant skin cell that now has these reprogrammed genes in it as well as the original DNA in it. Okay, and what these genes are reprogrammed to do is to turn the cell into a pluripotent cell. So it takes a already differentiated cell. So this cell has already decided it's going to be a skin cell, and it's going to actually turn it back into, it's going to go backwards and turn it into a pluripotent cell. That's the induced pluripotent part. Okay, and they're inducing it back into this pluripotent stem cell. And so now that it's been induced back into the pluripotent stem cell, okay, we incorporate the factors in that will make it differentiate into whatever other type of cell it needs to be differentiated into. So this example took this skin cell, incorporated the reprogrammed genes in it, so it induced it back to its pluripotent state, and then the factors were added to turn that actually into a nerve cell. And so this is ongoing research. Nobel Prizes were awarded for this in 2012, okay, for working with these iPSs. Okay, so they're not currently, being used to treat things yet. They're still in research. But that is one of the things we need to realize. All of these genetic engineering techniques that we talk about, they are constantly changing. They are like I said, these people just got Nobel Prizes in 2012. Okay, research is constantly coming out. Innovations are happening. The technology is changing. So these things are changing right now as we speak. There are new discoveries being made in these fields. But by improvements in this induced pluripotent stem cell process, that could possibly negate the need for embryonic stem cells, which would remove a lot of the ethical debate and pushback about this kind of research. The last technique we're going to look at is, this is more of a lump of a couple of techniques, is the forensic biotechnology. And these are things that can identify people from DNA samples, identify criminals, to identify disaster victims, to determine biological parents. So forensic biotechnologies are used to basically to ID people or organisms, okay, from a sample. And so the first one we're going to look at is electrophoresis. And you've seen this before in a simplified manner. You've seen this before with the DNA banding, where you've compared the bands and seen if they have matching bands, and so this person had to commit the crime. So we're going to talk a little bit more about how those bands come about and how this works. So you can see here, we've got a gel set up there, picture on the left. Okay, and so you'll notice, so how this will work is there's the gel that the DNA is inserted in, and the gel is in a buffer solution. Okay, and there's a power supply that's added. So electrical current goes through this buffer solution and through the chamber. And so there is a negative side and a positive side. And the DNA is inserted on the negative side. Okay, so that you can see where the DNA would be injected to the wells here on the negative side. And the DNA is injected on the negative side because DNA actually has a negative charge. And so if DNA has a negative charge, you guys, from what you know about negatives and positives, the negative DNA is attracted to the positive side here. So that's going to help pull the DNA that way through the gel. And a negative and a negative, okay, they don't like each other. So that's also going to push the DNA. So you've got a combination of the negative charge pushing the negatively charged DNA away from it, and the positive charge on the other side attracting the negative DNA towards it. Okay, and so that'll help the DNA move through the gel. The gel itself is like a tunnel matrix, almost like going through like a forest. Okay, and so the gel itself is a series of these small little tunnels. Okay, and so the DNA has to fit and squeeze through these tunnels and work its way through the gel as it's being moved by these charges. And so the smaller the DNA molecule is, the farther it can travel. So you can see on the second picture, you've got longer molecules that didn't go quite as far, and the shorter molecules that were able to travel farther because they were able to get through the gel easier than the longer fragments that get caught up in the gel. So you can see here, looking at comparisons of these banding patterns and seeing how they're different from person to person. And so the DNA itself is not already in little pieces. The DNA does have to be cut into these fragments that will be of different lengths. Like I said, some will be shorter, some will be longer. And so this technique is going to require the use of those restriction enzymes. And so remember, the restriction enzymes cut the DNA at certain places. And so those particular sequences will be at different places in different people. And so it ends up with different fragment lengths for the different individuals that are cut, even with this exact same restriction enzyme. And so we get what are called restriction fragment length polymorphisms. And so which are abbreviated RFLP. Okay, and so these restriction fragment length polymorphisms, these are the differences between people. These are why we get the different banding patterns and that allow us to ID different people. These are, the fact that I may have a bunch of short fragments of DNA where one of y'all may have a mix of short and long. Okay, and so that's going to be due to these cutting with the restriction enzyme and everyone getting a different length. Another technique we can use is what's called PCR. PCR stands for polymerase chain reaction. I can't spell today. That's an L. And so a polymerase chain reaction is basically an amplification of the DNA. Okay, this is when we have a very small sample of DNA. You can only run gel electrophoresis if you have a large sample of DNA, like from a test tube, from a blood sample. Okay, and that's not usually what is available, especially if you're doing something to, like for instance, a criminal case, and they've left just a small amount of DNA behind at the scene. And so polymerase chain reaction is used to make thousands of copies of the original very small sample of the DNA. We target key regions of it. Okay, they're called a targeted sequence, and we make thousands of copies of this particular targeted sequence. And then once those copies are made, then those can be, now you can do the electrophoresis. Okay, so we make all these copies of this target sequence that's relatively short. Okay, the target sequence is going to be less than 20,000 base pairs, which I know doesn't